Good evening everyone. So, the last class we were talking some examples of biostatistics, right, where statistics is involved in biomedical engineering, right. So, today we will continue on that. Today we will start slowly introducing terms, as I said, up to 22nd being a holiday, okay. From 23rd you will see a difference, okay. So, we will today cover again some examples to motivate and then take it slower. Once the registration freezes on 22nd, then we will take it at a different pace, okay. So, some of the terms which in the last class also we discussed, if you remember, is about a population, a sample, and then what do we have there, a study design and a inference, right. So, can somebody tell me what are the terms, meaning of those terms? Let us start with the bigger one. What is a population? We discussed this in the last class. What is population? Give me an example of a population. Let us say we do a clinical trial. What is population here? Humans, size, quantitative 7 billion, somewhere around that number is my population, that is of interest to us, right. Not necessarily, a drug is designed for what. So, that is a humans is a very broad term, then it is not never going to end at all, 7 billion is too big. What is the drug designed for? Let us say if it is a neoplasm or let us say for a certain cognitive drug or whatever, then your population size decreases because you are targeting a subset of the human species as a whole, right. Now, you want to see whether the drug is effective for these people, okay. And in clinical trial, one of the thing is so we are, if you take about the one saving grace is if you target a medicine, right, develop a medicine or develop a drug targeting a disorder or disease, you will have a clear warning. What is it? You do not simply buy it and take it, right, only those it is intended for specific use. Have you seen the labels? It is intended for specific use and most of them to be administered under prescription from the doctor. So, if you look at that is where Benadryl cough syrup, good. So, Benadryl has quite a bit of what? Alcohol content. Codeine is even worse, okay. Codeine is also for like common cold, etc., okay. It is a over the counter medicine, OTC, they call it as over the counter. For us, for in India almost everything is OTC, most of the drugs are OTC, you can go and buy from a chemist, which is not possible in many of the western world. So, it is called over the drug, OTC, over the counter medicine, you do not need a prescription. But what is its intended purpose? It is for what? When you have a cold cough, that should be taken in certain quantities, okay. Now, drug abuse can happen, especially with codeine. Codeine is something equivalent to a addictive molecule, okay, which can actually lead to drug abuse. But is codeine responsible for it or the drug? You cannot sue the company for this purpose because it is a misuse of drug, okay. So, remember when we are talking about clinical trials or drug discovery, etc., the intended purposes should be very clear. Whom are we targeting? So, it is not the population here does not mean the entire population, but the population you are targeting, that should be very clear. This is not just true in clinical trial, any experimental studies one has to be very clear about the population. Do not take it very generic, then you may never be able to prove the efficacy, prove it is working, it will always it will result in throwing away your hypothesis, okay. So, population is, you have to very clearly define the population, okay. So, if you ask remember I do not know, Alfonso mangoes very popular. 2 years back Alfonso was actually stopped by EU. Do you know that? European Union stopped importing Alfonso because it is of lower quality or whatever reasons, health issues. Okay. So, now what is the population there? The entire set of Alfonso mangoes being exported, not the entire set, which is being exported from certain place or certain country, I mean in this case India, right. And now what is the inference they had? They came up with the conclusion that they made a decision not to export. Remember, it's actually a tug of war, it is actually a prestigious moment for India as well. Okay. In fact, in light of it, some of the politicians went ahead in the EU meeting, they gave Alfonso as a gift. Okay. So, it is actually, it serves political issues as well, decisions. So, now how could one come up with this decision? Why would EU come up with this decision? You actually sample certain portion of the mangoes, right. So, if you think about quality control, so this, remember, it is not just bio, of course it is applied to bio, we are talking about some applications of bio, but what I want to say is it is very generic. Okay. So, in this example you are talking about a quality control case, equivalent to a device, a drug or a mango, quality control, right, and statistics plays a very important role in quality control. So, if I am exporting 1 million mangoes and I want to test whether it is good or bad, and it involves destructive testing, you understand what is destructive and non-destructive? Have you heard of? Today non-destructive testing is very popular, of course Professor Krishnan Balasubramaniam works in this mechanical engineering. Non-destructive is very popular, but sometimes you do not have an option to do destructive. So, suppose if you take a mango and start piercing, that is destructive. Why? You cannot sell that mango, that is of no use, right. But if you take a metal piece and then pass it to an X-ray or whatever, then still I am not making any harm to the structure, that way it is non-destructive. So, many a times you may want to do a destructive testing, then what will happen? If I have 1 million, can I do destructive testing on 1 million? It is like in movies, you eat 60 idlis and say after that, no, none of them is good, does not make any sense, right. So, you start doing what? Start taking samples. Now, the key question is how many samples to take? What is my objective here? What is my objective here? So, to make this conclusion, so these are 3 terms, right, but to do inference, what is required? You have taken a sample, now you know what is a sample. I do not know how many to take, but you know what is a sample, a subset, right. Now, what will be my next step? What is my objective here? What is my objective? So, in this case quality control of Alfonso mangoes, what is the objective? Let us take a simple example, let us say you have an objective. What is this? I will write it qualitatively and then make it quantitative. Your objective is what? Not just to find quality, is that all? You will find quality and say okay, this is the quality, you will leave it and go, is that what you make? What is objective?
Number of mangoes that are good and number of mangoes that are bad.
Okay, you only take a sample, with that sample what can you tell about population? You will tell anything, what do you want to tell about population? You have taken a sample, let us say you have tested the quality, say you have found some X are good, Y are bad. So what is it you want to prove? Your objective as a quality controller is to what? Accept the batch or reject the batch? As a quality control or a testing, you are, how many of you are somewhere for joined IT companies or have some IT exposure? Information technology, I mean computer companies, software companies, nobody? What is the job of a tester? To fail the software. That is why a tester sits there. I worked on both in development and testing for some time. So, the key idea is to fail the software, not accept. So as a quality control, your objective is to do what? If you are a standards person, so if you are part of standards of, let us say biomedical devices, or if you are a standards evaluating the quality of clinical trials, quality of a drug, you know different teams operate. If you are a developer, if you are involved in developing a molecule, you cannot be involved in trials, you cannot even administer the trials. What I meant to say is you cannot administer the trials, because you are involved in developing the drug molecule, you will not be involved in administering the clinical trials. Okay. Why? You will be biased, human tendency, you will be biased and say it is okay, you will try to defend your molecule, right, defend your baby, that is not going to be possible. So that is very clearly set aside. Okay. So what is, as a quality engineer, we have the objective is to reject the batch. Okay. So now if that is the case, somebody has developed it, somebody has put hard efforts to develop it, let us say mangoes, you know, it is a year of long effort, and any decision will actually strain the relationship between countries. Remember, it will strain the relationship between, these are big decisions, and any drug molecule everywhere. So imagine Pfizer's drug, if drug you know, there are many countries which tried. Why did you, how many of you took Sputnik? Why? There should be a reason why. Sputnik was also available.
Clinical trials were not, the sample size was not disclosed.
You see, if you read it carefully between the lines, you will know what is clinical. So what is a good study? You have to, when you say a drug molecule, you have to publish. How many of you have an Apple Watch? Have you looked at Apple Watch claims? So they say they have tested on some 600,000 patients and they were able to get an accuracy, high accurate heart rate. Now if I want to measure heart rate, will I use, you know, Apple Watch given an option, or will I use you know, our Xiaomi? Which one will you prefer? Quality. Where does quality come from? Stats, testing, stats. You have to make sure, that is where things are different. That is where things, why do you say you are looking up to a paper in Nature compared to paper in some XYZ journal? Simple, you cannot publish in Nature without large scale studies, right. So the value add that a statistics gives is huge, a proper study, right. So that is where, so you, if you want to prove something, okay, here I want to prove that I want to reject the batch is bad, okay. Now how are you going to do this? So I want to reject the batch. How do you set about yourself? Now your objective is clear. What is it? I want to reject the batch, or you want to, you know, in a drug molecule, what will be the objective? I will tell you why, okay. For in case of a drug molecule, you will say it will serve the purpose. I will tell you why later on, okay. So this objective, I will tell you why an objective has to be carefully chosen, okay. So in this case, it is reject the batch. Now remember, rejecting the batch, is it going to be tough job or easy job? Is it, do you want to reject the batch with high confidence or moderate confidence? Remember, 100% is not possible. Why? Why 100% is not possible? Huh? You are only working on sample. So 100% confidence is not possible. Forget it, because you are working on a subset. Clear everyone? There is no doubt about it. 100% is not a possibility. Now you have only a sample, with sample you are inferring. Now if I want to reject this mango batch, should my confidence be higher or lower? Why? Because it is a very critical decision, right. It will strain relationships, and not only that, let us say I have been importing for a long time, all of a sudden 1 year you come and say that it is not working, I can sue you in the courts, right. So better be proper when you reject. So you have to put a lot of efforts to reject the batch. Now you understand why my null hypothesis, why, no, I will not bring in the jargons, why my objective in clinical trials is prove it is working, because you have to put lot of work to make sure that it is working. You should have make sure you achieve the objective with high confidence. So whatever you want to prove or disprove with high confidence, that is your objective. I do not say it is negative or positive. Sometimes you want to take a negative as the objective, and sometimes you may want to take positive as the objective, but keep in mind the objective is something you want to be, outcome you should be very sure about. That is the way you want to even keep the objective, okay, you want to be very sure about. So now how do I make it as a qualitative statement? Quantitative statement. How do I make this as a quantitative statement? Reject the mango batch. What do I do? How do you say a mango is good or bad? That is the question. How do you say that? So there comes set of parameters of interest, right? You may have a single parameter or a vector of parameters, right. So slowly we will get into math sooner. So you will have a single parameter or vector of parameters. What will be these parameters usually? What will be these parameters? So let us say in clinical trial, what do you want? Take a simple example, tell me what it is. Let us say if somebody comes up with a drug to cure common cold, relieve common cold, how do I test that? Side effects is negative. First let us start with positive. Huh? How well is a qualitative term, right? What is quantitative? Time taken to recover, okay. Then in COVID vaccines what is the case? Antibodies, you have to measure the antibodies produced, right, that is the measure, right. Now on top of it, so on top of it what do you want? Antibodies may be X for some person, Y for some other person. So you want to say an average, I will not use math symbols, I will come back to it, average of parameter value for sample. So on an average how much antibodies were produced after administering the vaccine, clear? That is one thing. So you have to find a set of parameters that will make sure that is of interest to you first, that will ensure that if the set of parameters are let us say greater than or less than certain number, you say that this is pass or this is failed. So you have to have that idea. So this is like this is converting a qualitative objective to a quantitative objective. So when you talk about vaccine, you talk about antibodies. When you talk about recovery, when you talk about a drug molecule, you say what will you test for? Let us say a drug molecule for neoplasm, a cancer, what will you test for? Average number of cells died, good cells died versus bad cells died versus the good cells also. Why is it important? Then I would say target specificity. How am I able to go to hit a particular target, not the entire, right. So if the specificity is higher, it is a great thing. So you will list a lot of things, not just efficacy, specificity is another thing, right. Any drug molecule, without, that is where interactions come into picture or side effects come into picture. does it interact and create side effects? What is the side effect of plasm is more killing of normal cells. I mean, testing the radiation therapy, you will result, it will result in killing of normal cells as well. You want to have that reduced. So is it specific to a particular target? What else? What else you can think of in drugs? Leave aside mango in this case. Let's think about drugs. What are the examples you can think of? What are the parameters? Vaccines you think about antibodies, then specificity, how many cells in cancer case, some other example. Quantity of drug. Dosage, that is a dose, it is a dose issue, how much dose I should increase, right. But for X dose, dose is a factor. So what are we talking about? In other words, set of parameters we call it as, in jargon we call it as factors, factors which will influence your output. These are called factors. You will talk about 2 factorial experiments, multifactorial experiments, etc. Okay. So here we are talking about factors. So what are the factors? Factors is dosage levels, okay, and what else you can change in the inputs. So these are factors. These are not the ones which we are output levels. These factors will influence your parameters of interest. Okay. So parameters of interest is still your quality term. Whatever you can change in input, that is factors. We will come to that later. So let us stick still some more example about parameters. Let us say a drug for what do we have? Oh my god, are we short of diseases? There are so many diseases, right? Pick up one. Diabetes. There is no cure. If there is anything, I would have been happy, right? Some bacterial infection, TB. What do you how do you say that TB medicine works? Because today that is antibiotic, right? So how do you test an antibiotic? How do you test an antibiotic? Samples, then do what? Swab is there. So what are we looking for? Either you take a sample or you take blood, samples, or you take a swab, etc. What are we looking for? How do you first know? So the easiest part, okay, is how do you diagnose? That is what you test for, right. But how do you diagnose a TB? Blood samples, swab, or you have this usual body check, right. Blood sample is there, urine sample, okay, fecal sample, you go on doing all of these samples. Either one of them people figure out the presence of bacteria, or for a specific organ you have this imaging systems, right. So now those are diagnostic tools. You use the diagnostic tool to know that, and in fact when you do this testing, what do? You again use this diagnostic tools to see how it has been reducing. That is all the efficacy is. So if you want to, you do not need to break your head to think about how you want to, what are the parameters to look for. Why? What is the parameter you looked for in diagnosing? That is the parameter you should look for in testing the drug as well. It is a very simple idea. Only for vaccines it is difficult because you do not look for those are non-destructive testing. You do not want to do destructive testing in humans, right, because basically you cannot expose it to a rabies and then say is it working, no, that is when you go for antibodies. So that is where testing itself is 2 types, destructive, non-destructive, right. So that is why I said target group should be very clear, the population should be very clear. You cannot have a whole population for your study. For vaccines, what is the population? Whole population probably above 18. Vaccine is one of the toughest because why? You cannot expose them to the real virus. We only develop mostly vaccines for virus, right. So you cannot expose it to a virus, but you want to see whether antibodies are produced, okay. So that is very different, that is kind of non-destructive. Whereas if you talk about HIV, if you talk about neoplasm cancers, if you talk about any kind of disease, even Alzheimer's, there are focused study groups. People call them as focused study groups, clinical trials, where you ask a patient to voluntarily come and join the group. Okay, they will be administered medicine for free, of course, and then you follow them closely to see how they progress. So your target group is very different. Population, you have to be very careful in defining. Once you know the population, then statistics will tell you what is the size, etc. And now that we understand how to make it into a quantitative problem, how will you make it into a quantitative problem? Simple. If there is a disease, then you look for how they diagnose the disease. Then you put a bar saying these are the factors that I should look for, and this should be going down over a period of time. The time factor is also important. Okay. So that will determine how efficient it is, how much is it able to cure, all of this. If you track those parameters which are used for diagnosis, you will make this problem into a quantitative problem. Then factors that influence the output. So remember, when you talk about drugs, a drug A together with B may have a very high influence. A drug A at different levels will have different influence. So remember, they say you that in a day how much how much mg of you can take of paracetamol, or how much mg of paracetamol you can take, in a day how much mg you can take of paracetamol? No idea. Even after COVID we do not have an idea. No idea. 2000 4 hours once. 4 hours once you can take, that is the worst case. So 6 is what you can take, 6 tablets. I know I have done all of this, so that is why I know. Okay, 6 does not help me, did not help me at that time. So 6 into 650, okay, the worst, somewhere around 3500. The content of paracetamol will be still lower, somewhere around 3000 is the limit, I guess. How did they come up? Side effects. So you have to look for factors that will affect your parameters both positively and negatively. Increasing the dose may have a quicker effect, but will have another. So have you heard of this pharmacology, study called pharmacology? That will determine. So there is a study called pharmacology which will actually tell you pharmacological parameters, like how much time it takes, what is the half life of the drug, how much time it takes to flush the drug out of the system, right. So these are some of the parameters one has to look for. So and these are also factors, the dose is an important factor. For example, factors influencing is dosage, dose levels, that is why I call this dosage level, that is a factor. At different levels it will have different interactions. And then multiple drugs, could be synergy, could be detrimental, right. So these are various factors that will infer, so interaction among drugs. Interaction among drugs at various doses, okay. So it soon becomes complicated, okay, factors. So you have an objective, qualitative, try to convert it into quantitative, then you look for factors, then what is it we do? We start with something called, before we do an inference. So this all comes under where? Study design, okay. You take a population, okay, and then even before taking a sample you have to enumerate all of this. Why? That is when you know what sample to even look for, how many samples to look for. Because if you are going to do at 10 dose levels, can I take only one sample from each dose? No, right? You need to have enough samples for each dose level, okay. So when you list your objective very carefully, then your study design will keep increasing. I mean your study design will tell you how much sample you should pick up, okay. So if your population, you have a certain population, your objective is to just reject, that is not objective, you have to convert it into quantitative. The moment you convert it into quantitative, you will know the issues, because you need to may need to monitor not just one parameter, a vector of parameters, okay, and you may need to monitor for gender, okay. So you don't want to avoid, you want to avoid biases, so you want to do it for different genders, different age groups, okay, and you want to look for different dose levels and interaction among drugs, known interaction among drugs. Then when you enumerate all of this, sample size will start increasing, the design will get complicated, okay. Then how do I start inferencing? So then we have what is known as a null hypothesis and a alternate hypothesis, okay. So that is where things are. So null hypothesis is something like a status quo. So in the case of mango you will say that, what will you say, accept or reject, status quo, accept, because you do not have enough evidence to reject it, accept. In case of drug molecules, what will you say, drug discovery, you will say this molecule does not work, that is the status quo. Now you have to put your hard efforts to with sample studies, with enough samples, with statistics to say that this is working. You have to disprove your null hypothesis, that is usually the case. You have to, with evidence, disprove null hypothesis. Okay, with evidence we have to disprove your null hypothesis. Keep in mind the statistics, I did not show you a slide, later on I will show you. There are many ways to lie, and one of the ways to lie is using statistics, okay. If you misuse statistics, you can produce great lies, okay, but that does not suffice the purpose, okay. Statistics have to be used carefully, interpreted carefully, very carefully. So if I ask you what is confidence interval, how many of you heard about confidence interval? So let us say so I do not know how many of you have known this, we will discuss this much later point. So what I have written is the true mean mu lies between these two intervals, and I am sure it will lie between these two intervals 95%, okay. This many, this has many catches, okay. First of all, this sigma, okay, we will come to this later. I think I will put you into a higher degree without going through the basics. Let us come back to this later. So when you talk about confidence intervals, the interpretation needs to be very careful. So why am I saying this? Let us say take a simple example to understand. So how many of you know about election polls, exit polls? Does exit polls always predict what happens, even in the recent election? Recent election, what happened? Give me a case where recent elections exit polls were bad and the results were different. I mean, exit polls were predicting something and results were predicting something. Karnataka was Congress. Close call. Something else. The recent ones, Madhya Pradesh. Madhya Pradesh was nowhere. They said it was a very close call, probably a hanged assembly, etc. Nothing of, yeah, but nothing of that sort happened, right? So what has gone wrong? Did the company make any mistake? The exit polls, did they make any mistake? Of course, when you do the truth, you can always say they were not correct, but the same thing that did they do it for other states? It worked. it did not work here. So what is it? Is it a hit and miss, or is there a scientific evidence, or is there something else? So sometimes they are correct, many a times they are correct, sometimes they are off, right. The last I think in the last elections almost most of the parties predicted, most of the exit polls predicted the current government will be the one which will come, but they were off in seats, margin was very different. Only one of them India Today I guess was very close to the margin what really the party got, around 260 plus, right. Many of them are saying 200, 220, somewhere very on the lower side. India Today was betting on 260, even I was surprised. Coalition, so nothing of that sort happened. In fact, they were much better off even before the first elections, right. So 260, yeah, somewhere a single largest party itself is 260. So I looked at it and said everywhere somewhere converging, this India Today guy alone had a very different prediction. Is there any difference in scientific approach? Do you think will there be any difference in scientific approach? Statistics cannot change, the approach, the framework cannot change, the framework is same, and it is not that they have been doing it for years, so it is not going to be that they will lose out on framework. What happened is the kind of samples they collected, kind of samples they collected. So when you say confidence, you have to take it with a pinch of salt. That is why I said you have to be very careful, because you can produce great lies using statistics. You can say you are confident 95%, but when you go and look back at the samples will be smaller in size. You can come to a conclusion with 100 samples, you can conclude conclusion with 1000 or 100,000 samples, and the more and more samples you take, your confidence interval is going to be this range, whatever I have written, mu less than this range is going to be tighter. You are going to closely predict your truth, you are getting closer and closer to the truth. Of course you will never know the truth, that is philosophical, and actually in theory as well we state the same, you will never know the truth, but you will get closer and closer to the truth, okay. So the overall flow is then you have a population, you make a study design, you have a sample, and then you start inferencing using statistics. Then why do we call it a statistics? Remember, we have a vector of parameters, but that parameter set is for one entry in a sample. Now your sample size could be of n, large number, let us say you take 10,000 or 100,000, for each entity you will have 10 parameters. Let us say if I am talking about clinical trial, for each person you would have measured what, 10 parameters after administering the drug. Now like that how many people you would have done? Let us say you have done 100, 1000 or 10,000, you will have a vector of, it is a matrix, you will have 10,000 cross 10 matrix, right. Now with that I want to make a conclusion, that is where statistics come into picture.